Okay, screen sharing is done, and let us open the WhatsApp. Okay. Chats, fine. Okay, so good evening, everyone. I'm very happy today that 63 of you have joined this class, and today we are going to start with Mendelism. Okay, so just clear me three things: the audio, the video, and the presentation. Are the three things visible to you all? Visible, audible?
Yes, sir.
Okay. Okay, fine. Thank you. My video is stuck or okay, fine. It's fine. Just a minute. I hope there is no background noise that is disturbing you, right?
No, sir.
Okay, fine. Thank you. Thank you so much. So I welcome you all to this ICAR JRF Plant Science classes. And this is the Master Course 2024 for the Plant Sciences JRF. And today we are going to start a new chapter, which is Mendelism, which is going to be very much interesting for you all. So before that, let me just again introduce myself. I'm Ariprasad SK, currently a PhD scholar from Indian Agriculture Research Institute, New Delhi, in the Department of Seed Science and Technology. So I have cleared the ICAR JRF exam with All India 48th rank during 2020, and I just took IRI for Seed Science. And then I have also cleared the IBPS mains in the same year. Later, I just joined as completed my master's, and I have also secured All India second rank in my SRF in the subject Seed Science and Technology. And I'm currently pursuing the PhD in the same institute, that is Indian Agriculture Research Institute, New Delhi. And recently, I have cleared the ASRB net in the 2022 in the same subject. So this is a brief introduction. So this is a brief introduction about myself. Okay. So let me just begin with today's class. So there might be few curiosity, like a few things that will have that from which you can get some curiosity about the Mendel, his work, and you might be curious enough to just read more about his paper or more about his work from the things that I will be going to share in today's class. Okay. So let us just begin the class with explaining about the Mendel, about his history, and regarding his wonderful piece of work. So back in 18th century, the actual condition was all the scientists like biologists, biochemists, everyone, they were in a race to find out the one major thing, which is the trait or the character. So you take any organisms, like everyone were well aware of the trait, the phenotypic trait that was visible to our naked eye. But the question was how these traits are actually passing from generation to generation. There were variations, there were alternative forms. Like for example, if I just take the quantitative one, height, there is a difference of height in when you just survey a group of people. This is in case of humans. If we take plants, in case of color, of flower color, seed color, anything, there is at least two different types of colors that we see. So seeing all these things, the students were scientists were actually curious about to know how this is transferring from generation to generation. What is the actual cause? So that time they already made a hypothesis or made an assumption that there might be some factors which is actually responsible for all these characters, and this is how it is transferring from one generation to another generation. But out of all the race, from all the participants, like Mendel actually won the race, but he couldn't get the recognition, or he was not able to prove his experiment which he has done, even after presenting it two times in 1865. He presented his piece of work for the two times in February 8 and also March 8 in 1865, but he was like none of the scientists, like none of the other scientists were in the condition to accept the theories or accept the laws or the ideas that were put forward by the Mendel. Okay, so this is just a brief history about what has happened back in 18th century. So we'll just go in detail and read about the Mendel, his life history, and the experiments that he has done in the several organisms. Okay, so we'll just start with the Mendel. So if I want to just talk about the Mendel, is the enigma. Enigma in the sense like is hard to explain, and also difficult to understand for whatever work that he has done, because at the same time he's highly praised, and also at the same time he's highly condemned for his work. That few people or few renowned scientists claim that he has cheated and he has just misnumbered, like he fabricated the data and he has presented it in the natural, like in the presentation that he did in the 1865. So these controversies are still there. So this is how it is. So you also get like you also get to know that he is the father of genetics, but at the same time there are also several other class of people who still condemn his work and say that he has fabricated the values and they were not actually done in the 1865, back in 18th century. Regarding the Mendel, so where was he born? Yes, these things, yeah, Czechoslovakia. So what is it now? So he was born in Moravia. So all these things are very much important. Make sure to remember these places. Okay. So it was Moravia in Brunn, which was in Austria. So what is it currently? It's Brno in the Czechoslovakia. Okay. So this is where the Mendel was born. What is the date? What was when did he born? In 1822. So what about the date? You can also unmute and answer. So it was in July 20, 1822, right? The place and the year are clear. Okay. So about his studies, so he left his studies in very early since he belonged to a very poor family. So he has to drop his studies and he joined as a priest in a monastery, which was a Augustinian monastery in the Brunn. And later he became a priest in 1847. So after becoming a priest in 1847, in 1851, he went to University of Vienna to study several subjects like physics, mathematics, philosophy, and etc. So after that, he returned to Brunn in 1854 and he was appointed as a substitute teacher for the subject science. So you will be amazed to hear this. Even after getting appointed as a substitute teacher, he failed in his Viva exam. So where he couldn't get get a job as a permanent faculty in the same institute, because he was not able to clear that entrance exam. That was mainly because of failing in Viva. So he was not able to get that job. So back after 1854, he came back and he continued to be as a priest in the same monastery, or the monk or priest in the same monastery. So this is in brief about the Mendel, or what he has done, about his studies and about his occupation, what he was actually doing. So he actually failed for 2 times in this Viva, and finally he came back to the Brunn and he has continued as a priest there. So after being as a priest, he just started the experiments, which we have currently accepted universal laws, which are called as Mendel laws. Right. So initially, talking about the experimental material that he has used, he started his experiment not with the bees, he started his experiment with the mice. But what made him to transfer his experimental material from mice to pea? Anyone has any idea about it? Or in the first place, I hope like you already know, like he started with mice, because this was even asked in 2023 paper, I guess. If you have already seen the 2023 paper, they have asked about the experimental material, which was the mice. Any idea like why he just shifted his experiment material from the mice directly to the plant, from animal to plant? So what made him to do so? So it was because of the difficulties that he faced in the monastery, where the monastery people didn't allow him to experiment with the mice, because since it was creating, so he was actually breeding the mice, so which was creating the disturbance in the surrounding of the monastery. So monastery people forced him. Yes. Tanya was answering it, like people from the monastery opposed to use the mice. Yes. So since they opposed and they forced him to stop breeding the mice, he just shifted his experimental material from mice to pea. So why he selected the peas? Can you just list out the reasons why he selected the pea as his experimental material?
Contrasting characters.
A contrasting character we can take even with the several plants, we can just select the contrasting characters and study. But what was the main reasons? So that is one of the reason. Yes, I agree with that.
It completes its life cycle for a short period of time.
It was a pure breeding or self-pollinated. First thing, the short life cycle, and you can also easy to
It
cultivate.
is bisexual.
Yes, sorry.
It is bisexual. It was bisexual.
Yeah, it is self-pollinated, bisexual. Okay, fine.
The
So
larger seed size.
yeah, larger seed size. And lastly, someone has told like it has several contrasting characters that he can easily observe. So these are the few reasons why he actually selected the P for his P as his experimental material. First thing, he just cultivated in this piece in his kitchen garden. He has to go nowhere for this cultivation or not to select a particular field. He did his complete experiment in his kitchen garden itself. Okay, so these are the few things that you should be able to remember if you are studying about the Mendel, about the place where he is born from, about his educational history, about his birth date, and also the experimental material that he has used and why he has selected that experimental material. So all these things will be very much important for you. Okay. So the Mendel, he is considered as a brilliant fellow even after failing in some exams. So there are several reasons for it. Can you just list out one or two reasons why he was considered as brilliant? If we just go through his experiment, you might come with some conclusions about him, or you can judge something about him. So why you think like he was brilliant?
Because at that time, statistical tools were not used in biology. He used them.
Okay.
That is why he was brilliant.
Not actually statistical method. Statistical method is a broader term. He just used a simple mathematics. Yeah, you are very close to it. Okay. So you use the mathematics to understand the transfer of traits from generation to generation. As like the amount of data that he was getting was increasing by generation to generation. So he correlated with it with the ratios, and this is how he was able to come for a proper conclusion. So this is the one thing why he was considered brilliant, because as soon as he came to know that the experimental material is getting the data which he is collecting is getting bigger and bigger, he simply applied the mathematics to it. So this is the one reason. So any other reasons? Studied only one trait at the time.
Complete
So okay.
control of F1 is not.
Yes, exactly. See, while he was preparing his experiment, he has done the proper analysis of what others were doing. So he has analyzed everything, since all other scientists were actually not concentrating on a single character or a single trait at a time. So they were not able to come into a proper conclusion. But seeing this lacuna, the Mendel selected only a single, not selected actually, he just focused on a single trait. He did not bother about the other traits from generation to generation. He mainly focused a single trait. He was observing what was happening to this trait for several generations. So this is how he outnumbered, or this is how it's something new that he has thought. Okay. So this is the second thing, analysis of the work, and third one, selecting a single trait. So what else? Someone was saying something.
Kept the record.
Yeah. So he kept the complete record. So he kept the complete record. That is one reason. And also, he actually perfectly designed the experiment and named them. For example, he went from parents to F1, F1 to F2, F2 to F3, and this is how he named his generations. there was a proper naming so that there was no confusion at all from generation to generation, right? So this is one of the reason. And so he was extremely careful. So that is very much needed. So all these are the reasons. And anything else? Okay. So I was talking about the mathematics, where he was able to convert all these things into a simple, all the data into simple ratio, where this was the main reason why he was able to come into a proper conclusion, because he turned everything into the ratios. So he was able to come into the proper conclusion. Okay, fine. So this type of things might not directly help you in the exams, but if you know all these things, the background stories about him, so you'll be able to definitely answer if they are asking something very different from what they are given in the book. Okay, so make sure to listen to all these things very carefully. This is how you get the interest in the subjects. When you try to know the things outside the box or outside the book, so this is how you will get the curiosity, right? So starting with his experiment, how he started with P. So we are with the P now, like from the mice, we have come till P. So how he started his experiment with the P? Can anyone just tell me like how he started it and how many number of varieties he selected, and out of which finally how much he derived from it? If you have the numbers. So it was
34,
in
sir.
It was in 18, yes, very good. It was in 1857, he started collecting the seeds of the pea. So he started collecting the peas from the commercial growers, where he selected 34 pea varieties, out of which he bred them for 2 years. After 2 years, he selected 22 varieties which were performing stable with least variations. Okay. So chose 34 varieties, selected 22 among them. Okay, very good. So these 22 varieties he selected, and in these 22 varieties, he selected easily scoreable traits, which simply means the quantitative traits. So this was something about his experiment. The experimental material was, as I said you, it was the Pisum sativum, and they were easily grown in the kitchen garden. So this is very important about his experimental material, why he selected the P. They will definitely directly ask you in the either assertioning reasoning or the statement based questions. So make sure to know all these things, all these points. Okay. Next. Okay. I'll just ask you one question. Just tell me like whether you feel Mendel was extremely lucky or everything is complete hard work.
It was his hard work.
Hard
Okay.
work and patience.
No, sir, Mendel was lucky.
Okay. There are two different types of answer. Okay. I'm getting
Lucky,
some.
sir. Lucky.
Yeah.
Not
Both?
complete. Yes.
Okay. I won't use it or, let us just plus it. Add it. Okay. Lucky plus he was hardworking. Because of his hard work or because he was brilliance or the skills, you can take everything. So this, but whatever you say, the luck was a factor which was with him while he was doing the experiment. So I'll just tell you one simple thing here, what has happened actually. Forget about all the things that you have learned from the book. Okay. So there is one thing that you might be not known about. Have you heard about the P wheel? You might be heard about it in the any entomology class. So what has happened, like he concluded his experiments in 1863. So in the very next year in 1864, this pea will destroy the complete crop in the Brunn area. So Brunn area. See, this was something that he was very lucky enough, because if his experimental material was destroyed by this pea wheel, he couldn't come into the proper conclusion. So he, even after his conclusion, like concluding his experiment in the immediate year, like this pea wheel destroyed the complete area of Brunn pea plant. So this is why, like, I feel is extremely lucky after reading about this thing. So what are the other things that you might be read from the books? Yeah, he did not encounter linkage. Okay, 1 point. What are the other things?
Having
So
monogenic control, sir. All the characters which are selected by.
yes, very good. The 7 traits which the Mendel selected were under monogenic control. So is this completely true? All the 7 traits that are listed by the Mendel are under monogenic control. That is, single gene is responsible for that trait. The characters we choose were monogenic. Yes, that is what Vishwaja is answering. Okay, fine, good. What do you think? All 7 traits were under monogenic control. Is it
Except
100%?
seed color, sir.
So seed color. So what is the nature of the seed color? It is under polygenic control or any other phenomena. Yes, just think about it.
By 2 characters, two traits, red and yellow. नहीं, red and कुछ green है. Green, yeah.
Every trait that is selected as 2 alternative forms. Okay, let me just clarify this thing. So yes, they were all under the monogenic control. That was the first thing. So whatever traits that he selected, they were under monogenic control. So that was the second point, or the third point, why he was lucky enough. So what I did not encounter linkage, that is fine. So these are the 3 important points that you can take while you justify that Mendel was lucky enough since he didn't encounter all these conditions. Okay. So talking about the 7 traits that okay, let us just talk about the other organisms first, which E worked on, and then let us come back to the P and the 7 traits. So can you just list out the other organisms that E has worked with, if you remember?
Honeybee, heartweed, visual.
Okay. Honeybee, very good. Hawkweed. Yes, Rajma. Physalis. Okay, Physalis, Rajma, fine. So these, and let us take it as mice and then Pisum sativa. Okay. The series, it was from mice and Pisum, P, and he came to some conclusion, and then he tried it with all these other organisms. But in none of these organisms, he was able to come to the same conclusion that you could come from the P. Like working with P, the conclusion that he is derived, the same conclusion he was not able to derive from all these 3 plants or 3 organisms you can take. So what is the reason? So mainly because the male were actually haploid and female were diploid. So what about the haploid?
Parthenogenesis.
It was mainly the apomictic. Yes. So what is apomictic and how it is different from parthenogenesis? What is
In apomixis, only male parent contributes to the embryo.
not actually male parent? You cannot tell like that. No, it's not a male.
Male.
Seed without fertilization.
Okay, seed without fertilization is parthenocarpy. Even in the apomixis, the same thing happens, I guess. It's without the fertilization, the apomixis. So what is the difference then? Okay, do not worry about it. We'll discuss this in detail in the seed technology classes. I can assure you, like, all your doubts will be cleared. Okay, so coming back, it was awkward. Why was not successful? Because of apomixis. So what about Rajma? Why was not successful? Sorry. No answer.
Pure line.
Okay, you just search it and just tell me tomorrow. Okay, this Rajma one. So mice, you already know, they didn't allow him to carry out the experiment. That is how it is done. Fine. So Pisum, you already know, it's since it is pure breeding, and there are several other things or characters that made him to select this plant, and he was successful in deriving some conclusion. Whether the conclusion that he derived was true or false, that is a different thing. But at least from the material, he was able to come to certain conclusion. That is very important. Okay. So coming back, I will just talk about the paper first. So he presented his paper for the first time in 1865 at 2 different dates. So February 8th and March 8th, the same date in the different months. So it was at the meeting of what? at a meeting of Society of Natural History Society. Society of Natural History of Brun. So this is where he presented his findings. Later in the next year, it was in 1866, his paper was published. In which journal? It was in Annual Proceedings of the Society. So what was the language of this paper, if you remember? In which language did the Mendel wrote the paper? Yes, exactly. Very good. It was in German language, and interestingly, it was completely handwritten, and totally it consists of 48 pages. I'll just show you the paper, the first page of the paper. So this is the paper which Mendel wrote. It was published all over the Europe and the USA. And most interestingly, he also sent his paper to Charles Darwin, but since he was there in the same time, but he didn't read the paper at all. Forget about giving some thoughts about the paper. He didn't even read the paper because he was not okay with the idea that he applied the mathematics to the biology. He was not okay with the ideas, so he didn't even read the paper. So this was a few things about the Mendel's paper, how it went. So what you should remember from this is the dates 1865, where he presented his paper and where it was published. So what about the title of the paper? So what was the title of the paper? The
So
title of the,
experiments in plant hybridization.
yes, experiments
Experiment in plant hybrids.
who is this plant hybrids? Who was that? Plant hybrids. Karthik. Okay, you listen. You
Experiment in
attended.
plant hybrids.
You attended the previous year class.
Yes, sir.
Okay, fine. So it is actually the experiments in plant hybrids, but nowhere you will be seeing this as hybrids. It is hybridization. But recently, in one symposium for the bicentenary, like 200 birthday of Mendel, there was one symposium at IRI where BD Singh himself told this. The correct translation of the German language is the experiment in the plant hybrids. So this is just for your knowledge, but in the exams, if they are asking you, like they will be either giving the hybridization, mostly the hybridization. They won't confuse you with the hybrids and hybridization. Just stick to it. But at least know the thing that it is actually the experiment in plant hybrids. If you are interested, you can just ask me for the link. I will just send the YouTube link. There is a one YouTube lecture by BD Singh that was given for in the occasion of bicentenary of Mendel. So there you can clearly see what he has told about the Mendel's title. Okay. So as I mentioned, also like once he published his work, all the reprints were sent all over the Europe and USA and also to the several scientists. So most of them, like the main reason why it didn't get popularized was because of use of mathematics. No one was willing to accept these things. So that is why the Mendel work didn't come into existence at all until he died. So it was next. One more important thing to remember is it actually cited for 3 times after his death. After publishing his paper, before till 1900, it was cited for 3 times. So how many of you understand about this cite? Like how many of you know about what I mean by cite? Citing a paper.
Rediscovery.
Citing a paper, it seems, for example, if I just write anything about certain topic, for example, I just write about certain chemical, I'll take my seminar topic. I just write about something about the coumarins. Coumarins are the chemical compounds which are phenolic compounds which will have some positive effect in seed germination. So while writing it, where do I collect the information about the coumarins? So where do I do it? So I do it from the literature that has been already published, right? So I take some information from there and just I prepare from my own. So that is what citing means. If I take some information from other papers or the other literature, I have to cite the names of the author. So that is what citation means. And here it was cited only 3 times before it was rediscovered in 1900. So these are something outside the box, like outside the book. but it's interesting. Okay. So later it was rediscovered. As you all know this thing, it was after 16 years, it was rediscovered in 1900 by 3 scientists independently, right? So I already named the 3 scientists. Hugo de Veres from Poland. It was Erich von Schermach and Karl Korans from Germany, from Austria. So these were the 3 scientists who independently came into the same conclusion. When they search back the literature which supports their conclusion, all the 3 scientists came to a single paper, which was the experiments in plant hybridization, which was published by the Mendel. So this is how the Mendel's work was rediscovered. And one more interesting thing, out of these 3 scientists, the Erich von Schermach and the Karl Korans actually praised the Mendel work. They actually praised the Mendel work, but Hugo de Veres was not happy with giving the title the Mendel, the father of genetics. He was not happy with the fact that the complete credit was given to the Mendel. He just said that it is very unfair to give him the credit. The credit should be given to us. So this is what he said. But the other 2 scientists were actually supporting the work that was done by the Mendel. Okay. So these are the things about the Mendel's paper. Okay, and we already discussed about the word genetics. It is geneticus, which means to generate or genesis, the origin. So this paper. So this is a book written by whom? Who was the first person? Or let me just take the name itself, Bateson. So what is the important contribution of the William Bateson? If you can.
Translated Mendel's works.
Okay, he was the first person to translate the Mendel's work in 1902 after the rediscovery from 3 different scientists. So he is the main reason why actually the Mendel's paper got popularized all over the world, because since it was in the German language, not many could be able to read the language. So only after its English translation, which was done by the William Bateson, so this complete Mendel's work got hugely popularized mainly because of his contribution. So this is the book originally written by the William Bateson. So this is a very nice book. You can also read it. I have also not read it completely. I just came to know about this book a few days before. So this is completely available in the internet. You can just go and read it, okay, if you are interested. So a lot of things we have discussed, rediscovery. So while we talk about the rediscovery of the Mendel works, there is also one more scientist who is Spillman, W.J. Spillman. Some also credit Spillman for the independent discovery, but this is not mentioned as per the records, as they are mentioning these 3 different scientists. But some even credit him also for the same thing. Claim of honor, I already talked about it. So Mendel's work contributed for clearing 2 important things that was very much confusing back in date. The first thing was regarding the animalculist and the oist, which I talked in the previous class. While I was talking about the theories of evolution, I told you about the 2 different classes or 2 different categories of people, right? How many of you remember them? The animalculist and the oist. So this was in which one? The pangenesis or preformation?
Pangenesis.
Yes, it was in pangenesis. I told you about the 2 different categories of the people who actually, the first class of people mainly believed that it was a sperm from which the progeny will come, and just the female egg just acts as ovary, just acts as the nutritive tissue. The oist, they believed that it is mainly because of the ovary or the egg cell, and the sperms only provide the stimulatory effect, right? So this confusion was finally clarified by the Mendel's work, where he concluded that there is always an equal contribution of the gametes from male and female. So this was the first controversy which was clarified by the Mendel's work. And the second thing was the heredity material. So about the heredity material, which I was talking about, was the factor where the Mendel used the term factor to explain the heredity material. So this factor was not known to be completely a particulate. So before the Mendel's work, so you should understand this, before the Mendel's work, so this was not completely known to be particulate. They were also using the term called as blending inheritance. So where the gametes from female and male will mix with each other, and this is how we get the new trait. So is it true now, the blending inheritance, that the gametes that are passed from the males and female will be blend with each other, and this is how we get the trait in the progeny?
No, sir.
No,
Yeah.
sir.
Because the factors which the Mendel was referring to were actually particulate. So there is no such blending inheritance. So that confusion was finally clarified with the Mendel's work. Am I clear with the things that I have explained till now? So how many of you have felt that there is something interesting that you have learned today? Just raise your hands. Yes, be quick. Okay, very good. So let us just come back to the traits that I was talking about. The 7 traits that were studied by the Mendel. So where there was someone who was quoting this seed color, there is some confusion with the seed color. I will just clarify this confusion. Okay. So let me just, you people just list out the 7 traits first.
Seed shape,
Okay. Let us go one by one. Seed
And
shape,
Seed color,
Seed code color.
seed color,
pod color,
cotyledon color,
cotyledon color,
plant height,
Pod color.
pod shape,
plant, pod shape,
position of
plant height,
plant height is odd,
Length of stem.
plant height is odd, length of stem length,
flower position,
pod color, yeah. Lastly, it is flower position. So did I miss anything? Seed color you can take, or seed coat color. Seed coat color on a cotyledon color. So did I miss anything? Just check it once. Seed shape, seed coat color, cotyledon color, plant height, pod shape, pod color, flower position. Okay. Akshita is mentioning about the flower color. So where does this flower color fit into? Is it the 8th trait that Mendel studied, or it should fit it somewhere in within the 7 traits?
sir, seed
Yes.
color, seed color and pod color. It is, I feel, in these two.
Pod is the complete one. So don't get confused with the seed color and pod color. They are completely different. So where should this flower color fit into?
cotyledon color.
Yes. So it is not a cotyledon color where the seed color should fit into. Sorry, yeah, cotyledon color is where the flower color should fit into. Why it is so? Why I'm just writing the 2 different traits for a single gene? Let us take it as a single gene. So there were total of 5 genes. So these 7 traits are actually distributed over the 5 genes in the P. So what does this mean? If I just write the cotyledon color or the flower color, what did you understand by this? You might be seeing this in every paper or in the every book that you see. They will be either mentioning the flower color or the cotyledon color or the both by mentioning it as or. So what does this mean? Just think about it. It's very simple. I already given you the int, the 5 genes. Yes, anyone? So let me just show you the figure. So the cotyledon color, okay, the flower color and the seed coat color. I just wrongly mentioned it, I guess. It's not the seed coat color. The cotyledon color is a different. It is a seed coat color or the flower color. Sorry. The cotyledon color and the seed color are same. Seed color and cotyledon color are same, and it is the seed coat color or the flower color. White and purple color genes for the flower. So what does it mean? Like, I didn't get your point. White and purple color genes for the flower means? White and purple color genes for the flower. So my simple question is why they are denoting it as OR. The simple reason is these are completely different traits. It doesn't mean that both are same. These are completely different traits, but the thing is both are controlled by a single gene. So the gene is showing the pleiotropic effect. How many of you have got this point now? Just raise your hands if you have understood what I am explaining. If not, just ask me like if you're not able to understand. So these things you won't be getting from the book. If you just ask your seniors, if you just ask some doubt to your professors, then only you'll be able to know it. So this is how we are actually knowing it, just by discussing with our friends, juniors and seniors. I hope you got the point. Why it is OR. So if someone will come and ask you, some of your friends who have not taken this course, if they are reading by themselves, they will definitely get this question. Why it is actually mentioned it as flower color or seed coat color, or only one thing is mentioned, which one is correct? They might be also thinking that the 7 traits that are mentioned in the like that Mendel worked were different in different books. They will just think that okay, this is been misprinted. This should be something else. But I hope like you know the correct reason now, right? Yes or no?
Yes.
Okay, so the flower color and the seed coat color, it was controlled by a single gene, a gene which is showing the pleiotropic effect. So in case of flower color, it is purple and white. In the seed coat color, it is gray and white. So white remains the same, but here the color differs. The flower, it is purple. The seed coat color, it is gray. So talking about the chromosome numbers, where they are actually located, this were the old one. So old published dates or old published genes, gene number. So this is in 711445. So I used to remember it as 1457 while I was preparing for my JRF. But it recently got revised, and the new numbers or the new genes where they are located, or new chromosomes, sorry. So this is the chromosomes. The new chromosomes where these genes are located, genes which are controlling these traits are located, has been revised. If you want to remember, you have to remember this new chromosome numbers, not the old one. Okay. And if required, also try to remember the symbols which are used to denote these traits, like capital R small r, I small I, A small a, FA small fa, or FAS small fas. It is LE small le, and P small p, V small v, and lastly it is GP small g. Okay, so these are the 7 different traits the Mendel's on which the Mendel worked on. The flower color, no, like he mainly worked on the seed coat color. Okay, so one more interesting thing. So out of these 7 traits, which are actually present in how many genes? How many genes? Just count the genes. It is 1, 2, 3, 4, 5. All the genes. So my camera might be masking the numbers, but it is 1, 2, 3, 4, 5. So there are how many genes total? Total 5 genes contributing for 7 different traits. So out of these 7 different traits, the 4 traits have been completely cloned. So we have the complete information regarding the 4 traits. So what are the traits? The traits are the stem length, the seed shape, the seed coat color or the flower color, and the cotyledon color or the seed color. Is it clear? So you can expect this question in the upcoming JRF exam, probably. So make sure to remember it. Not genes, like a traits. Okay. So this is about the 7 traits that were studied by the Mendel. So let us just start with the monohybrid cross. So what he has done and what conclusion he has derived from this monohybrid cross. So simply the explaining his experiment, what he has done. He has just taken one single trait, for example, the plant height. So he just collected the tall plant and crossed with a single dwarf plant. So in the next generation, all were tall. So what he observed, the F1 were all tall. So again, he self-did. So, but in the F2 generation, he got tall and dwarf. So he got this in a variable number. So since he applied the mathematics, he converted this into the ratio, where the ratio was 2.84:1. So this was his findings from the monohybrid cross. First of all, he just considered it as 2 different alleles, right? For example, T will be capital T and small t. So this is how he has explained that every factor has an alternative form. That will be the one factor will be capital T and one other factor will be small t. That is, one will be dominant and one will be recessive to it. So why he has considered only 2 different alleles here? Why not 3? Why not 4?
Because it is a monogenic trait.
So monogenic, okay. So monogenic in terms like, what do you understand by monogenic?
Single trait.
Yeah, like single gene
One
contributes to
gene control is the
trait.
one character.
One
Yeah.
trait. So then why he has taken the 2? There will be 2 alternative forms of every gene. How he came to know about it? Yes, your point is okay. Like this is monogenic. I completely agree with it. So why he has assumed that okay, all the genes will have the 2 alternative forms, right? So this is what he
Because
has
he found only 2 plants, tall and dwarf.
exactly. So since he only got 2 different types in the 2 different varieties for the trait, for a single trait, which is plant height here. So it was tall and dwarf. So what he assumed was tall is because of some factor and dwarf is because of some factor. So this is what he assumed. So these are the 2 different factors. He assumed it. But in the F1, what he saw was all were tall. So there he explained that the one factor is actually masking the other factor. Right. So the one which is masking, he gave it a term, the expressing factor, and the one which was actually masked, or one which was actually recessive by the name we know it today, so he named it as a latent factor. So this is how he explained his monohybrid cross. So explained it like, so there will be pure breeding in terms, it will be always TT, which will be crossed with small t. So in the F1, since there will be exactly a small capital T and a small t in all the plants, so one will actually mask the other, so we'll get only one type of phenotype. So that would be either tall or dwarf. If we are getting tall, that means the capital T, the factor which is responsible for tall, is masking the recessive one. That is, it is dominant. So this is the conclusion. So after in the F2, since it is reverting back, he gave the ratios. He just crossed the capital T small t into capital T small t. As per the mathematical calculation, it should be capital T, capital T, capital T small t, small t, small t. The ratio should be 1:2:1. If in case both the gametes or both the factors are contributing equally. If they are contributing equally, this should be the ratio in the F2. So while comparing it with the mathematical calculation with the actual observation, he found the same thing, where the ratios actually matched. So that is how we came to the conclusion that each gamete, like each factors, contribute equally from males and female. So this was his monohybrid cross in a simple way. Have you understood it?
Yes, sir.
Okay. I know like many of you already understood these things from reading the book, but I just tried to explain in a bit different way. And a similar experiment he conducted with all other all other 6 traits, and he came to the same conclusion. If we just talk about the ratios, and these were the ratios. For the one which I showed, this was 2.84, and for the rest, they were the numbers are actually changing. That's it, but they are always near to 3. And he also gave a statement that this variation is because of chance. This is just a chance error why their number is not exactly 3, right? So pure breeding in terms of they might have missed because of some outcrossing might have been done in the plant's pollens. The foreign pollen might have come in that, or instead of self pollination, the pollen from the other plant have come, and that is how it has got fertilized. So this is why the numbers are not exactly 3. Okay. So there are certain terms which we can define before going further. So the terms are alleles. The first term would be allele. So how do you define this allele? The allele is nothing but the alternative form of a gene. So who gave this term allele?
Johnson.
Bateson.
It was the Bateson. Don't get confused with the terms. That is why I'm making the list. I know this is where most of the students will be confusing. So make sure to remember it from now. So next, genotype. So how do you define this genotype? Phenotype. Next, I'll just list out. You can just recall and tell me. Yeah, Aishwarya sent one gene has 2 alleles. Okay, I didn't read your message earlier. Sorry. Yeah, you are right. Yeah, what does this genotype means? Genotype is just a allelic constitution, whether it is capital A, small a, capital A, or small a, small a. So allelic constitution of organisms or each strain would be the genotype. So who gave the term? It was by
Johnson.
Yes, it was by Johnson. You people are bored of answering, or you people are getting bored of just listening to the things that you already know. It was in 1909. So what is phenotype then? It is just a physical appearance of each strain. That would be the phenotype. So what is F1? F1 is derived from the Latin word, which means the son or the daughter. So that is what F1 means. F means filial. Filial is derived from the Latin word, which means the son or the daughter. So who gave this term F1 or filial? It was by Bateson. Okay, remember this.
Bateson.
Yeah, very good. Tanya is answering. Fine, good. So gene is nothing but the basic unit of inheritance. It was again given by Johnson. And homozygous and heterozygous, they were also given by Bateson. So the homozygous is nothing but the 2 identical alleles. For example, capital A, small a, small a. So heterozygous is nothing but the different alleles of a particular gene will be the heterozygous one. So these are the important terms that you should be remembering along with the scientist. I hope there would be no confusion from now on, right? So let us just propose one law based on what we have discussed about the law of which one? The monohybrid cross. That is law of segregation. So what does this law of segregation tells us? They do not mix the segregate. The gametes will come into F1 and they will again segregate from next generation. This is what, in simple terms, this law of segregation means. They won't mix with each other. The alleles won't mix. They will be separated from each other from generation to generation. So the F2 ratio for the monohybrid cross will be 3:1 and F3 ratio will be 1:1. And what is the other name for this law of segregation? If you remember?
Low purity.
Yes, very good.
Pure.
Speak
Law of
louder.
purity of gametes.
Be confident. So it is law of purity of gametes. So it's pretty much important that you also remember the alternative names for it, because in the exam every time it's not very sure that they will only give this law of segregation. They might be using the law of purity of gametes instead. So be sure to know all the alternative names for a single word. Okay. So this is law of segregation. That is fine. So I was telling you about the equal frequency of the gametes. So what are the evidence? So how can we know that both male and female are actually contributing in an equal way or equal fashion or equal proportion? So how do we get to know this? So we have two different things. One is indirect evidence we can take, or there is also a direct evidence. So indirect evidence is by the test cross. Very good. Ramya has answered it. So test cross is mainly used for two purposes. So what are the two purposes? If you can list it out.
Can find the genotype. Homozygous or hetero.
First thing is to knowing the proportion or the whether the F1 has the equal frequency of males and females. So that is the one application of this test cross. And what is the other one? So other one is to know the genotype of a strain. To know the genotype of a strain, we can use this test cross. Fine. I'll just show you here. So consider that if you are crossing the heterozygous with the homozygous recessive plants, the offspring will be 1:1. When it will be 1:1? If you are crossing the heterozygous with homozygous, when it will be always equal to 1:1. When there is an equal contribution of all the gametes. Just assume that there is equal contribution, only then you will get the 1:1 ratio. So this is how it is the indirect evidence for the equal frequency of the gametes. Then what about the genotype? For example, I have a certain plant which is actually growing tall. So what can be the genotype of this plant? There are two possibilities, right? Am I right? There are two possibilities for this tall plant. The genotype can be of two possibilities. Is it true or not?
Yes.
Yes, sir.
So what are the possibilities? It can be either capital T or capital E small t. So how to identify this? How to know whether it is capital T or small t? Simply what you need to do is you just cross this tall into known dwarf plant. So dwarf is obviously the small t. There is no other option here. It should be always equal to it should be always small t only. This is why it is dwarf, right? Because it's recessive in nature. Only if it is in the homozygous condition, then only it would be dwarf. It would express the dwarfness. So we already know this, know the genotype of this plant, that is recessive plant. That is what test cross means, right? So we just cross F1 into the recessive plant. So that is the definition of the test cross. So if I cross the tall into dwarf, if I get only tall, so what does it mean? So what will be the genotype of this plant then? If I'm only getting the tall in the F1 generation, it's TT. But what if I'm also getting the dwarf with tall? So it simply means that it is capital T small t. So this is a simple explanation regarding how we can identify the genotype using the test cross. Am I clear?
Yes, sir.
Okay.
Yes, sir.
So let us just list out the exceptions of the first law. So which is very much important in the exam point of view. So just list out the exception for the first law which you have learned now, which is law of segregation. So first thing, it should not be cytoplasmic. There should be no cytoplasmic inheritance. So that we will discuss in the later class. You need to worry about it. So there are terms called as penetrance. I will just go back here only. So it is cytoplasmic inheritance, phenocopy. I will explain this in a later class. Don't worry. The penetrance, expressivity, the transposable elements, the term transposable elements, and there is one study called as epigenetics. This is also an exception, and the mutation and the polyploid. So all these are the exceptions to the first law. If any of these phenomena is occurring, then the law doesn't hold good. So, is it clear? About phenocopy, penetrance, expressivity, epigenetics, polyploidy, mutation, I will be discussing in the future classes since we have a separate chapter for all these things. No need to worry about it. So there is another other evidence, which would be the direct evidence for the same thing, for telling the equal frequency of gametes. So do you want me to explain this, or you already understood the thing? How many want me to explain this? Just raise your hands. Or if you have understood it, we can go forward. Okay. So this is another simple experiment to understand. You have to know the reason what we are doing. So we are trying to find that whether there is an equal contribution from males and females or not. So identify this. this experiment can be used. So they have just taken one gene, which is the waxy gene. So what is the function of this waxy gene? So this will, see, normally the maize plant will produce this deep blue color when it is treated with iodine. The pollen grains which have a gene which is WX, so it will stain deep blue when it is treated with iodine. So this is the wild type. So, but if it is in mutated or if it is in recessive form, so then in that case, it will give brown color when it is treated with iodine. So this recessive gene was identified, or this recessive allele was identified to be waxy gene. Okay. So in the heterozygous conditions, if I just give a heterozygous condition, so this is small wx, capital WX. If you have gone through the microsporogenesis, you would be understanding the formation of pollen grains. So this is diploid now. So what will be the ploidy level of pollen grains?
Haploid.
So it is haploid. So it is the result of meiosis, right? So the diploid will undergo meiosis, and this is how the VR gametes are formed. So the gametes, it would be, the gametes would contain the WX and also small WX, right? The capital WX and small WX. Small WX is nothing but waxy. So for example, there is a plant, so it is releasing the pollens. So there is a heterozygous plant which is releasing the pollens. The pollens are huge in number. Let us take the 1000 pollens have been released. So these pollens will be used to pollinate the plant, the recessive plant you can take. So when it is this heterozygous pollen grains have both the WX and small x, so these are used to pollinate the plant. Counting the pollen grains from the heterozygous plant, not even pollinating, let us leave it. For example, so now it is producing 1000 pollen grains. This heterozygous plant is producing 1000 pollen grains. If they are staining these pollen grains and watching, like observing this in a microscope, so what they have observed is they got equal proportion of brown stained pollen grains and deep blue stained pollen grains. So what does this mean? If, for example, from the 1000 pollen grains, if you are getting 500 pollen grains which are giving brown color when treated with iodine, and other 500 is also giving deep blue when treated with iodine. So what does it mean? There is an equal proportion of the gametes that are produced. Is it right or not?
Sir.
Have you understood the thing or not? Should I explain it again? Raise your hands if I want to explain it, if you want me to explain it again. Okay. Okay, let me just explain it again. Just a second. See, there is one heterozygous plant. So the heterozygous plant means it has a capital WX, small WX. So let us discuss the function of capital WX. So this is a gene which is responsible for producing deep blue when treated with iodine. This is in the pollen grain. Okay, we do know that pollen grain as the haploid. Either it will have capital WX or the small WX. So the small WX, so if this is there, this will give the brown color instead of deep blue when treated with iodine, right? But in the heterozygous, both will be there in the heterozygous condition. But if it is forming the pollen grains, that means every cell will have either capital WX or small WX. So once the pollen grains are formed, let us take that 1000 pollen grains are forming. So out of this 1000 pollen grains, when they were observed in the microscopes after staining, they observed the 500 which are actually producing the brown and other 500 which are actually producing the blue color. So this is a direct evidence from which we can say that there is an equal frequency of gametes for each allele. For both the alleles, there will be the equal frequency of the gametes that will be produced in the pollen grain. So this is one of the direct evidence. I hope this is clear. And there is also one more, I think, one more evidence for regarding the same. It was regarding the neurospora. I think you have missed it. So you can also see the similar kind of example with neurospora in case of spores. You can just read it. You can easily understand that. I won't be explaining it. It will simply just consume the time. Okay. Next, we have the second law of the Mendel, which he proposed. It was the law of independent assortment. So where this is a dihybrid cross. Why the name dihybrid cross? Because he has taken two traits at a time. So they were yellow and brown seeds with the green and wrinkled seeds. So in this case also, he has got a single uniform trait in the F1, and again it got segregated in F2. And this was comparable to the monogenic, like monohybrid cross. So based on this this observation, he came to the conclusion that the genes or the alleles of two different genes do not mix with each other. They will be separated after the gamete formation. That is why you are clearly seeing yellow with round, yellow with wrinkled, green with round, green with wrinkled, all four possible types. If there was improper mixing, that this ratio should not have come. But since there was no blending, they are not mixing with each other, they are segregating independently or assorting independently, you are seeing this ratio. So this is what he came into conclusion after doing a dihybrid cross. Along with it, during the dihybrid cross, he also mentioned this thing that each trait again is controlled by the different gene. So that is why we are getting the ratio as expected. As we have expected in the monohybrid ratio, the similar ratio is coming in F2 dihybrid cross because of the point that each trait is bound by the different gene. So they are not mixing with each other, they are starting independently. So that is why we are getting this ratio. So this was his conclusion from the independent assortment. So again, talking about the assumptions and all, you might be already knowing it. So talking about the exceptions. What are the exceptions of second law? If you can list the exceptions for the second law, what are they? Everyone got bored or what? Bored of knowing the things that you already know.
Linking.
Okay, this should be the first point. The linkage is the main exception to the second law. If there is a linkage between the two genes, you cannot get the ratio as expected in the F2 for the dihybrid cross. So this is the main limitation or the main exception you can take. Not limitation, exception. And next it is epistasis. It is gene interaction. The interaction between the two genes will modify their appearance or the trait, the way they are expressing. So the epistasis is another exception. And all other exceptions of Mendel's first law you can take. And also the pleiotropic. The pleiotropic effect will also disturb this assortment. So this is also an exception. And fourth, we have the maternal effect. So all these are the exceptions for the independent assortments, which are very much important. Try to remember, also try to know why these are the exceptions. So linkage, epistasis, anyhow, we'll be discussing in detail in the upcoming classes. Pleiotropy, maternal expert, everything will be discussed. We'll be understanding like why they are actually the exceptions, why it won't work if these phenomena are there in any of the trait. So am I clear the things I have explained till now?
Yes, sir.
Okay, so I have a few questions for you. Before that, I have listed the formulas. So these formulas are very much important. These are just memory based. You have to remember it. So there is also logic behind all these formulas. If you know the logic, it's good. But otherwise, at least remember the formulas directly. Okay, I have made the complete list of formulas for the gametes here, starting with the different kind of gametes for F1, different F2 phenotypes, total number of different F2 phenotypes, total number of different F2 genotypes, total number of F2 combinations, and total F2 genotypes that are homozygous, total F2 genotypes that are heterozygous. So you can definitely expect one question from these formulas. to make sure to remember it after after I upload the notes. Just download it and recall these things from time to time. Okay. So let us just start with the questions that were from the yesterday. Yesterday I forgot to add the questions. So these are from the ICAR JRF previous year papers. So this will actually revise the things. We can revise whatever we have discussed by just attempting these questions. Just answer, like, give me the option, which option is true out of these four. They are just stating to arrange the organelles from center to periphery. That is center to outside.
Sir, second option.
Okay, what will be the first one? The nucleolus. Okay. After nucleolus, what will be the next one? So it is endoplasmic reticulum, then Golgi bodies. Okay. And it is the plasma membrane. And lastly, it is cell wall. Okay.
Cell wall.
Fine. Yeah. Next. Four. Okay.
Fourth option.
Okay. The main product of photosynthesis is what? It is
Glucose.
Okay. The energy carrying molecule that cell uses for the energy. It's ATP. Okay. The organisms that make their own food, they are autotrophs. Okay. Then lastly, the organelles for organelle of photosynthesis, it is chloroplast. The option is D. See, these are the very simple questions that you can encounter in the exams. So make sure to make no mistakes in these questions. There are students who will actually make the mistakes while, because in the actual conditions, you might have to scroll down to see the options. The match the following will be in the upper side. You have to scroll down to see the options. So in that time, students will be confused and they will be giving the wrong options. So what I suggest is, as soon as you see the match the following, just write the things in the paper, the piece of paper that they will give. Just mark the things like ABCD. Like, for example, you'll just write ABCD quickly. You just mark it and just give an arrow mark and just try to match them. So if you are doing it in your mind, there many a times you will definitely miss a mark in it, which are very simple questions. So don't make this mistake. Next one. Rish, okay, 1, 1, 1, 2. Both statement 1 and statement 2 are incorrect. Why it is incorrect? It is correct. Okay. So pre means like prior to 1940, it was believed that genes were considered to be the Beatson stream. That was right, and it was not divisible. So this was a thing that was assumed before 1940. Only after 1940, the Oliver proved that genes are divisible. Okay, so both the statements are right. So this is something related to history that is I have taken. Like anyhow, we'll be discussing it in the molecular genetics again. While not in the molecular genetics, while discussing the complementation test, we'll again go through this complete experiment. Okay, so I already got the answer. Lysosomes 2.
Second option, mitochondria.
Okay. Why lysosomes? Debabrata, why it is lysosome? So I think I have not told you about this thing, right? Semi-autonomous. Which are the organelles which are semi-autonomous? Can you list them?
Chloroplast, mitochondria.
So okay, very good. So
Have
why
their
they are?
own D-
So why
Have
they
their
are?
own DNA.
own
Okay.
DNA.
So own DNA, that is why they are semi-autonomous. If they are having their own DNA, they should be called as autonomous. Why semi-autonomous? If they can replicate their own genetic material and if they can survive of their own, then they should be called as autonomous. But why semi-autonomous? You people are getting my point.
So half functions depend on their cell.
Sorry, please pardon. What did you say?
Sir, half function depend on their cell.
Their function depend on their cell.
Half function.
Not exactly off. The thing is, okay, at least you have tried. Very good. The thing is, they cannot completely serve. Yeah, Lohit.
No, sir. Sorry, carry on, sir.
Okay, it's no problem. We can answer. Like whenever you answer, that is when you remember. Majorly, whatever you answer, will be remembering them forever. What I say, it requires the frequent revision for you to remember. So why they are semi-autonomous? For the reason that they cannot be completely independently survive within the eukaryotic cell. They need some assistance from the nucleus. Why nucleus? The proteins or the enzymes that are synthesized from the genetic materials from the nucleus, they need some support for their survival. So that is why they are semi-autonomous. Why the term autonomous is used? Because they have their own genetic material. They have the capability to produce their own enzymes and proteins needed for the survival. But the thing is, they require the assistance from the genes that are present in the nucleus. They cannot independently survive on their own. So that is why the name semi-autonomous. Explaining this concept, have you just got some theory in your mind? Have you just remembered some theory in your mind that is related to this mitochondria and chloroplast? There is one theory that I didn't tell you in the previous class in the ultrastructure. Yes, Lohit, you want to answer? Or Janani, you are unmuted. You want to say something? So have you heard about the endo-symbiont theory? None of you have heard about this theory, the endo-symbiont theory. Yes or no? At least just say yes or no.
No, sir.
Okay. So this is one important theory. Yukesh has heard it. Okay, fine. So this is one important theory. So when we talk about the origin of mitochondria and chloroplast, see, we basically get this common question: why only these two organelles have this DNA? How come? How come these cell organelles in the eukaryotes have the DNA? How it is so? So when we just study the evolutionary process, so these two are believed to be a prokaryotic cell initially. Okay, so they were the independent prokaryotic cells like bacteria. They were bacteria itself, but somehow they came into the eukaryotic cell where they started living as the symbionts, deriving the nutrition from the host, and then they got evolved where they lost most of their function. But since the genetic material was there, that was not eroded. So genetic material was still there, but they lost the most of the functions which are required for their independent survival. So that is how they become the endosymbiont. Endo in terms like inside. It is a symbiont inside the eukaryotic cell. So this is what the endosymbiont theory explains us. And that is how they become semi-autonomous. They didn't completely lose their function, but they lost their major function, which is required for their independent survival. So that after that, they have become dependent on the nucleus for the proper functioning of these two organelles. So this is just a theory which we believe that how this mitochondria and chloroplast have been existed. Am I clear?
Yes, sir.
Okay. Yeah, Mansi has answered it. Very good. So they originated from the prokaryotes. Actually, the chat section is in the different screen, the different monitor. I lose the focus while I explain the things. Okay, don't mind. So next question. So this is also very similar to the previous one which I showed you. See, this is how the questions are actually repeating from year to year. So you should not ever ignore solving the previous year papers. So let us just go from interior. So what is the first thing? It is always nucleus. Fine. What after nucleus? So it is endoplasmic reticulum. Okay, or it is very Ankit has answered it as 4. How it is 4? A should not come there. Three. Okay. So it is B, E. Fine. So what after E? So D will be the last. That is for sure. So this next one will be the C, and next it will be the A. So the gap between the cell wall and the plasma membrane. So this is a perinuclear space. Just remember it. Okay. So the answer would be 3. Am I clear? Just tell me whether whether I'm clear or not first.
Yes, sir.
Yes, sir.
Yes, sir.
Oh, next one. Mitochondria. Yeah, just take some time, read it properly. Don't be in a hurry.
Second option.
Yes, sir.
Second option. Okay, let us just start. So the mitochondria, what is the main function of this mitochondria? It is oxidation of the carbohydrates, lipids, and the protein. So this is how the ATP is generated, right? Next, that is why they are called as powerhouses of the cell. Okay. So next is endoplasmic reticulum. So what is the function of this endoplasmic reticulum, which I explained yesterday? It is the provide the controlled passage of mRNA from nucleus to cytoplasm. So what about lysosomes? Digestion of intracellular substances and foreign materials. And lastly, Golgi campus, it's just a packaging organelle. So the option would be the second one. So these are some simple questions that definitely they will be asking you in the exam, where you should not lose any marks. You should not take any chance. No, not even by chance, you have to leave it. Okay, you have to very carefully answer all these questions. So they are just like a boon for you, all just match the following kind of things. Ribosomes are made up of, or discuss this. Yes, they are made up of RNA and protein. Please don't keep no confusion in these things. The composition, which I have already emphasized a lot of times. So subunit, even this I have emphasized. So they have simply asked the ATS. In the upcoming papers, they might be splitting this and asking what are the components of 50S. Just tell me about the proteins and the RNA which are associated with it. So you should be able to answer this question also. So make sure to prepare it accordingly from the table that I have given you in the notes. Okay. So here the answer would be the B, the 40 and 60S. So whatever we have discussed in the class, in the cell ultrastructure, the theories of evolution, the history, and the Mendelism, you can clearly see the questions have been asked on from all these parts, right? Next one. Chloroplasts are the presence in the cells of. Whether it is B or C. It should be B and C. You have blue-green algae where they have this chloroplast pigment, right? It should be B and C only. So these are the things where the most of students will be confused. They will be knowing it, but they will be failing in interpreting the things. So make sure to give some time for these kind of question and interpret or guess correctly, or come to a proper conclusion based on the facts, not just by the guess. The answer is already here. So this is the one thing that we have discussed in the history of genetics, right? So why it was Nobel Prize? I asked you the same question, why it was awarded to only 3 these scientists. So none of you have answered it. See the importance. James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin were contributed significantly, and we have discussed in detail what are their contributions, right? But instead of this, the Rosalind Franklin was not awarded with the Nobel Prize. So they are just giving the options. Just read the options what they have given. So this was given in 2023 paper, I guess. This was in the 2023 latest paper because of the fact that she died before the result of Nobel Award, and the Nobel Awards are not given posthumously. They should be live in order to receive this Nobel Prize. Next question. So this is again the similar kind of statement which was asked before last year, I guess. So this, see the reason. This is so too funny. Like it cannot be shared than more than 3 persons at a time. So the answer is 3. The statement 1 is actually correct. She was not awarded. Like James Francis Crick, Maurice Wilkins were awarded. Rosalind was not awarded, not because of 3 or more, because she was died. She already died before the announcement of this Nobel Prize. So the last, I guess this would be there are last 2 questions. Yeah. Match the following. See the formula which I told you. I have made the list of the formulas. They have asked the same formulas. So these are just the kind of goons who have actually registered our course in the previous years. So direct questions have been come where we have clearly discussed all these things. So number of individuals in a perfect F2 population, what is it?
2 की power n.
Yes, it is
2 की power n.
2 to the power n or 4 to the power n?
So 2 the power n.
2 to the power n. Okay. Okay, just take a screenshot of it. And we'll just go back to that question again. We should answer it now again. Have you taken the screenshot? No. Should I go back again? Okay. Just take a screenshot of it and then compare it with the question. Okay, if you are done. Oh. So just answer me, like number of individuals in a perfect F2 population, what it will be?
4n.
It is 4n. Okay. So the number of perfect homozygous individuals in a F2 population. Why you people are not able to tell even after seeing the things? It is 2n. Okay, it needs some revision. Fine. Just go back and revise them. That you have already taken a screenshot. Anyhow, in the notes, it is already there. You just take a printout of the notes that I will be uploading soon in an hour, and then just revise from time to time. You can just stick it on a wall that you can see regularly so that you won't be confused. Okay. So number of different heterozygous genotypes in the F2 population. What is the option?
4.
4. So number of
2 की power.
Yes, 2 to the power of n. Very good. So number of different kinds of genotypes in a F2 population. Different genotypes. It is always 3n. Clear? So this is the one. Okay, there is another question. Okay, this is last question. I hope this part is clear, right? If you're not clear, you can just go back and just read that notes. You'll be clear with the things. But make sure, see, this is how they are asking the questions. Even the formulas are being asked in the match the following. The last one. The concept of gene is not static but dynamic. Arranged a changing concept of gene chronologically. If they are not mentioned it, okay, fine. Oh, wait a second. Sorry. Okay. I'll just name them. A, B, C, D. B, C, D, A. Okay. B, the isolation was for the first time for Frederick mixture. Okay. And then what happened after that? It was one gene, one metabolic block, and then it was one polypeptide, and lastly it is a proper definition of gene. So what is the currently accepted definition of a gene, if you remember? My camera got finished. Yes. So what is the currently accepted modern definition of a gene? So this you have to tell me in the next class along with the Rajma one. Why it was not successful in the Rajma and what is the modern definition of a gene, which is now accepted. Okay. So these are the things that I wanted to discuss. See, you have already seen like how many questions have been come from these three chapters: history, cell ultrastructure, and the Mendelism. So this is the importance of these chapters, even though it looks very simple, but you need to have a deep understanding about all these concepts. Only then you'll be able to answer these questions. I think like many of you who have not registered or registered our master course have joined this class. If you feel that these classes are interesting, you are learning something new, if these classes are helping your preparation, then definitely think of joining our master course. Definitely can improve your preparation and boost your preparation level. Okay. And also I would request unit of inheritance composed of segment of DNA. This is not that simple. I'll be giving the definition tomorrow, but at least do some homework. Okay. So I would also request all of you who have registered our master course, who are the members of our community. I would personally request you all to please share about our master course to all your friends. Just suggest our course. Just tell them to watch our YouTube videos or attend the free lectures. All these free lectures will be uploaded in the YouTube. If you definitely feel that these classes will definitely boost your preparation, to keep it going, we need some encouragement in order to sustain. So that encouragement, I already told you in numbers, we need more and more registration since we are more in number. So the kind of energy that you are seeing, in me, if you want to see the same kind of energy until the end of this course with all the other tutors, definitely we need some encouragement. This is the ultimate thing. See, in the late night, every day we are spending this time, and I used to prepare all this presentation all day, and I have to prepare all these things. This requires a lot of time. But listening to the class, it will take only 1 to 1.5 hours. You can save 1 to 2 days or 1 week of your time just by listening to this 1 hour class. So this is the importance of this online classes. So if you're feeling it really useful, I would just request each and everyone to please share about our online master course to your friends. I know like there will be several students who are actually preparing for, so many students who are actually preparing for the ICAR JRF plants and subjects especially. So I want each and everyone, at least tell them about our platform. It's up to them whether they want to buy the course or not. Okay. And I was actually thinking, I was waiting if someone will upload these things in your status, in your WhatsApp status. See, this is how you can market our course, or how you can promote our course. And promoting our course, we are already also giving some benefit for you. It's not free of cost. You are not doing it for free. We'll also provide some incentive from which you can at least buy some books or aid your preparation. So that is why we come up with the idea. So at least after saying this, if you cannot directly go and tell someone, at least you can upload these things in your status, so that if your friends, if they are preparing for the same exam, they will definitely approach us, right? So this is the kind request from my side for our efforts, that you are already seeing it on a live basis. So these are the live classes where we have to sit for complete day just to prepare the content and schedule it and present it, so that present it in a way that you will be satisfied. Otherwise, again, there is no point in it just doing these classes, right? So it's again a request. Please share with everyone, each and everyone who are actually preparing for the plantains. If you cannot directly share it with it, at least share in your WhatsApp status. I will be more happy to see the WhatsApp status of you all if you are really liking our courses. Okay. And thank you all. And this will be the last demo class. And from tomorrow onwards, there won't be any Zoom link. you have to directly open the app, and from there itself, from at 10 p.m. you will get a notification to join the class. You have to join the class, and we will be directly starting the class. Nothing else, no advertisement, nothing. I'll be just interacting with you people. You can directly interact with us, since you all the registered students will be there. No one outside will be present in the class. So feel free to start discussing with me, and we'll be definitely enjoying these classes. I can assure you that. Okay. Thank you. Thank you so much for attending. We'll meet in the next class. Bye-bye and good night. Thank you. Thank you so much. I hope, like, I expect at least one registration from each one of you. Already few people have already done it. Some have even done it like 3 or 4 registration, up to 5, 6 registration from a single event. I really appreciate it. But at least I expect one person from each one of you. That will definitely help us. So this is how you can thank us. Okay. Thank you so much for attending. We'll meet in the next class. Let us make these sessions much more interesting and much more useful for you all people. Okay. Thank you so much.